Unit 4. Plant Physiology. The description of structure and variation of living organisms over a period of time ended up as two apparently irreconcilable perspectives on biology. The two perspectives essentially rested on two levels of organization of life form and phenomena. One described at organismic and above level of organization, while the second described at cellular and molecular level of organization. The first resulted in ecology and related disciplines. The second resulted in physiology and biochemistry. Description of physiological processes in flowering plant as an example is what is given in the chapter in this unit. The process of mineral nutrition of plant photosynthesis, transport, respiration, and ultimately plant growth and development are described in molecular terms, but in the context of cellular activities and even at organism level wherever appropriate. The relation of the physiological processes to environment is also discussed. Scientist Melvin Calvin, born in Minnesota in April 1911, received his PhD in chemistry from the University of Minnesota. He served as professor of chemistry at the University of California, Berkeley, just after World War II, when the World War was under shock after the Hiroshima Nagasaki bombing and seeing the ill effects of radioactivity. Calvin and co-worker put radioactivity to beneficial use. He, along with J. A. Basham, studied reaction in green plants, forming sugar and other substances from raw materials like carbon dioxide, water, and minerals. By labeling the carbon dioxide with C14, Calvin proposed that plants change light energy to chemical energy by transferring an electron in an organized array of pigments, molecule, and other substances. The mapping of the pathway of carbon assimilation in photosynthesis earned him Nobel Prize in 1961. The principle of photosynthesis, as established by Calvin, are at present being used in study on renewable resources for energy and materials and basic study in solar energy research. Chapter 11. Transport in plants. Have you ever wondered how water reaches the top of tall trees, or for that matter how and why substances move from one cell to other? Whether all substances move in a similar way in the same direction, and whether metabolic energy is required for moving substance. Plants need to move molecules over very long distance, much more than animals do. They also do not have a circulatory system in place. Water taken up by the roots has to reach all parts of the plant up to the very tip of the growing stem. The photosynthetes or food synthesized by the leaf have also to be moved to all parts, including the root tips embedded deep inside the soil. Movement across short distance, say within the cell, across the membranes, and from cell to cell within the tissue has also to take place. To understand some of the transport processes that takes place in plants, one would have to recollect one's basic knowledge about the structure of the cell and the anatomy of the plant body. We also need to revisit our understanding of diffusion. Besides gaining some knowledge about chemical potential and ions, when we talk of the movement of substance, we need first to define what kind of movement we are talking about, and also what substance we are looking at. In a flowering plant, the substance that would need to be transported are water, mineral nutrients, organic nutrients, and plant growth regulator. Over a small distance, substance move by diffusion and by cytoplasmic streaming, supplemented by active transport. Transport over longer distance proceed through the vascular system, the xylem and the phloem, and is called translocation. An important aspect that needs to considered is the direction of transport. In rooted plants, transport in xylem of water and mineral is essentially unidirectional from roots to the stem. Organic and mineral nutrients, however, undergo multidirectional transport. Organic compounds synthesized in the photosynthetic leaves are exported to all the other parts of the plant, including storage organ. From the storage organ, they are later re-exported. The mineral nutrients are taken up by the roots and are transported upward into the stem, leaves, and the growing region. When any plant part undergoes senescence, nutrients may be withdrawn from such region and moved to the growing parts. Hormones or plant growth regulator and other chemical signals and are also transported, though in very small amount, sometimes in a strictly polarized or unidirectional manner from where they are synthesized to the other parts. Hence, in a flowering plant, there is a complex traffic of compound, but probably very orderly, moving in different direction, each organ receiving some substance and giving out some others. 11.1 Means of transport. 11.11 Diffusion. Movement by diffusion is passive and may be from one part of the cell to the other, or from cell to cell, or over short distance, say from the intercellular space of the leaf to the outside. No energy expenditure takes place in diffusion. Molecules move in a random fashion, the net result being substance moving from region of higher concentration to region of lower concentration. Diffusion is a slow process and is not dependent on a living system. Diffusion is obvious in gases and liquid, but diffusion in solid is more likely rather than of solid. Diffusion is very important to plants since it is the only means for gaseous movement within the plant body. Diffusion rates are affected by the gradient of concentration, the permeability of the membrane separating them, temperature and pressure. 11.12 Facilitated diffusion. As pointed out earlier, a gradient must already be present for diffusion to occur. The diffusion rate depends on the size of the substance. Obviously, smaller substances diffuse faster. The diffusion of any substance across a membrane also depends on its solubility in lipid. The major constituent of the membrane, substance soluble in lipid, diffuse through the membrane faster. Substances that have hydrophilic moiety find it difficult to pass through the membrane. Their movement has to be facilitated. Membrane protein provides sites at which such molecules cross the membrane. They do not set up a concentration gradient. A concentration gradient must already be present for molecules to diffuse even if facilitated by the proteins. This process is called facilitated diffusion. In facilitated diffusion, special protein help move substance across membrane without expenditure of ATP energy. Facilitated diffusion cannot cause net transport of molecule from a low to high concentration. This would require input of energy. Transport rate reaches a maximum when all of the protein transporters are being used. Facilitated diffusion is very specific. It allows cell to select substance for uptake. It is sensitive to inhibitor which react with protein side chain. The protein form channel in the membrane for molecules to pass through. Some channels are always open, others can be controlled. Some are large, allowing a variety of molecules to cross. The porins are proteins that form large pores in the outer membrane of the plastids, mitochondria, and some bacteria, allowing molecules up to the size of a small protein to pass through. Figure 11.1 shows an extracellular molecule bound to the transport protein. The transport protein then rotates and releases the molecule inside the cell. Example: water channels made up of eight different type of aquaporins. 11.1.21 Passive sympod and antipod. Some carrier or transport protein allow diffusion only if two types of molecule move together. In a sympod, both molecules cross the membrane in the same direction. In an antipod, they move in opposite direction, that is given in figure 11.2. When a molecule moves across a membrane independent of other molecules, the process is called unipod. 11.13 Active transport. Active transport uses energy to transport and pump molecule against a concentration gradient. Active transport is carried out by a specific membrane protein. Hence, different proteins in the membrane play a major role in both active as well as passive transport. Pumps are proteins that use energy to carry substance across the cell membrane. These pumps can transport substance from a lower concentration to high concentration, that is called as uphill transport. Transport rate reaches a maximum when all the protein transporters are being used or are saturated. Like enzymes, the carrier protein is very specific in what it carries across the membrane. These proteins are sensitive to inhibitor that react with protein side chain. 11.14 Comparison of different transport processes, that is given in table 11.1, gives a comparison of the different transport mechanism. Protein in the membrane are responsible for facilitated diffusion and active transport, and hence so common characteristics of being highly selective. They are liable to saturate, respond to inhibitor, and are under hormonal regulation. But diffusion, whether facilitated or not, takes place only along a gradient and do not use energy. 11.2 Plant water relation. Water is essential for all physiological activity of plant and plays a very important role in all living organism. It provides the medium in which most substances are dissolved. The protoplasm of the cell is nothing but water in which different molecules are dissolved and several particles suspended. A watermelon has over 92% water. Most herbaceous plants have only 10 to 50% of its fresh weight as dry matter. Of course, distribution of water within a plant vary. Woody parts have relatively very little water, while soft parts mostly contain water. A seed may appear dry, but it still has water. Otherwise, it would not be alive and respiring. Terrestrial plants take up huge amount of water daily, but most of it is lost to the air through evaporation from the leaves, that is transpiration. A mature corn plant absorbs almost 3 liters of water in a day, while a mustard plant absorbs water equal to its own weight in about 5 hours. Because of this high demand for water, it is not surprising that water is often the limiting factor for plant growth and productivity in both agriculture and natural environment. 11.21 Water potential. To comprehend plant water relation and understanding of certain standard terms is necessary. Water potential is a concept fundamental to understanding water movement. Solute potential and pressure potential are the two main components that determine water potential. Water molecule possess kinetic energy in liquid and gaseous form. They are in random motion that is both rapid and constant. The greater the concentration of water in a system, the greater is its kinetic energy or water potential. Hence, it is obvious that pure water will have the greatest water potential. If two systems containing water are in contact, random movement of water molecules will result in net movement of water molecules from the system with higher energy to the one with lower energy. Thus, water will move from the system containing water at higher water potential to the one having low water potential. This process of movement of substance down a gradient of free energy is called diffusion. Water potential is denoted by the Greek symbol psi and is expressed in pressure units such as Pascal. By convention, the water potential of pure water at standard temperature, which is not under any pressure, is taken to be zero. If some solute is dissolved in pure water, the solution has favor free water molecule and the concentration free energy of water decreases, reducing its water potential. Hence, all solutions have a lower water potential than pure water. The magnitude of these lowering due to dissolution of a solute is called solute potential or psi s. Psi s is always negative. The more the solute molecule, the lower more negative is the psi s. For a solution at atmospheric pressure, water potential is equal to solute potential. If a pressure greater than atmospheric pressure is applied to pure water or a solution, its water potential increases. It is equivalent to pumping water from one place to another. Can you think of any system in our body where pressure is built up? Pressure can build up in a plant system when water enters a plant cell due to diffusion, causing a pressure build-up against the cell wall. It makes the cell turgid. This increases the pressure potential. Pressure potential is usually positive, though in plants negative potential or tension in the water column in the xylem play a major role in water transport up a stem. Pressure potential is denoted by psi p. Water potential of a cell is affected by the both solute and pressure potential. The relationship between them is as follows. 11.22 Osmosis. The plant cell is surrounded by a cell membrane and a cell wall. The cell wall is freely permeable to water and substance in solution, hence is not a barrier to movement. In plants, the cell usually contains a large central vacuole whose contents, the vacuolar sap, contribute to the solute potential of the cell. In plant cells, the cell membrane and the membrane of the vacuole, the tonoplast, together are important determinants of movement of molecules in or out of the cell. Osmosis is the term used to refer specifically to the diffusion of water across a differently or selectively permeable membrane. Osmosis occurs spontaneously in response to a driving force. The net direction and rate of osmosis depend on the pressure gradient and concentration gradient. Water will move from its region of higher chemical potential to its region of lower chemical potential until equilibrium is reached. At equilibrium, the two chambers should have nearly the same water potential. You may have made a potato osmometer in your earlier classes in school. If the potato tuber is placed in water, the water enters the cavity in the potato tuber containing a concentrated solution of sugar due to osmosis. Let us discuss another experiment where a solution of sucrose in water taken in a funnel is separated from pure water in a beaker by a selectively permeable membrane. You can get this kind of membrane in an egg. Remove the yolk and the albumin through a small hole at one end of the egg and place the cell in a dilute solution of hydrochloric acid for a few hours. The egg cell dissolves, leaving the membrane intact. Water will move into the funnel, resulting in rise in the level of the solution in the funnel. This will continue till the equilibrium is reached. In case sucrose does diffuse out through the membrane through the membrane. Will this equilibrium will be reached. External pressure can be applied from the upper part of the funnel such that no water diffuses into the funnel through the membrane. This pressure required to prevent water from diffusing is in fact the osmotic pressure, and this is the function of the solute concentration. More the solute concentration, greater will be the pressure required to prevent water from diffusing. In numerically, osmotic pressure is equivalent to the osmotic potential, but the sign is opposite. Osmotic pressure is the positive pressure applied, while osmotic potential is negative. 11.23 Plasmolysis, the behavior of the plant cell or tissue with regard to water movement depend on the surrounding solution. If the external solution balances the osmotic pressure of the cytoplasm, it is said to be isotonic. If the external solution is more dilute than the cytoplasm, it is hypotonic, and if the external solution is more concentrated, it is hypertonic. Cells swell in hypotonic solution and shrink in hypertonic ones. Plasmolysis occurs when water moves out of the cell and the cell membrane of a plant cell shrink away from its cell wall. This occurs when the cell is placed in a solution that is hypertonic, has more solutes to the protoplasm. Water moves out. It is first lost from the cytoplasm and then from the vacuole. The water, when drawn out of the cell through diffusion into the extracellular outside cell fluid, causes the protoplasm to shrink away from the walls. The cell is said to be plasmolyzed. The movement of water occurred across the membrane, moving from an area of high water potential, that is the cell, to an area of lower water potential outside the cell, that is given in figure 11.5. What occupies the space between the cell wall and the shrunken protoplast in the plasmolyzed cell? When the cell is placed in an isotonic solution, there is no net flow of water towards the inside or outside. If the external solution balances the osmotic pressure of the cytoplasm, it is said to be isotonic. When water flows into the cell and out of the cell and are in equilibrium, the cells are said to be flaccid. The process of plasmolysis is usually reversible. When the cells are placed in a hypotonic solution, higher water potential or dilute solution as compared to the cytoplasm, water diffuses into the cell, causing the cytoplasm to build up a pressure against the wall that is called turgor pressure. The pressure exerted by the protoplasm due to entry of water against the rigid walls is called pressure potential. Because of the rigidity of the cell wall, the cell does not rupture. The turgor pressure is ultimately responsible for enlargement and extension of growth of cell. 11.24 Imbition. Imbition is a special type of diffusion when water is absorbed by solid colloids, causing them to increase in volume. The classical example of imbition are absorption of water by seeds and dry wood. The pressure that is produced by the swelling of wood had been used by prehistoric man to split rocks and boulders. If it were not for the pressure due to emission, seedlings would not have been able to emerge out of the soil into the open. They probably would not have been able to establish. Emission is also diffusion. Since water movement is along a concentration gradient, the seeds and other such materials have almost no water, hence they absorb water easily. Water potential gradient between the absorbent and the liquid imbibed is essential for imbibition. In addition, for any substance to imbibe any liquid, affinity between the adsorbent and the liquid is also a prerequisite. 11.3 Long distance transport of water. At some earlier stage, you might have carried out an experiment where you had placed a twig bearing white flower in colored water and had watched it turn color. On examining the cut end of the twig after a few hours, you had noted the region through which the colored water moved. That experiment very easily demonstrated that the path of water movement is through the vascular bundle, more specifically the xylem. Now we have to go further and try and understand the mechanism of movement of water and other substance up a plant. Long distance transport of substance within a plant cannot be by diffusion alone. Diffusion is a slow process. It can account for only short distance movement of molecule. For example, the movement of a molecule across a typical plant cell about 50 micrometer takes approximately 2.5 seconds. At this rate, can you calculate how many years it would take for the movement of molecules over a distance of 1 meter within a plant by diffusion alone? In large and complex organisms, often substances have to be moved to long distance. Sometimes the sites of production or absorption and sites of storage are too far from each other. Diffusion or active transport would not suffice. Special long distance transport system become necessary so as to move substance across long distance and at a much faster rate. Water and minerals and food are generally moved by mass or bulk flow system. Mass flow is the movement of substance in bulk or in mass from one point to another as a result of pressure differences between the 2 points. It is a characteristic of mass flow that substance, whether in solution or in suspension, are swept along at the same pace as in a flowing river. This is unlike diffusion, where different substances move independently depending on their concentration gradient. Bulk flow can be achieved either through a positive hydrostatic pressure gradient, example a garden hose, or a negative hydrostatic pressure gradient, example suction through a straw. The bulk movement of substance through the conducting or vascular tissue of plant is called translocation. Do you remember studying cross section of roots, stems and leaves of higher plants and studying the vascular system? The higher plants have highly specialized vascular tissue, xylem and phloem. Xylem is associated with translocation of mainly water, minerals, salt, some organic nitrogen and hormones from roots to the aerial parts of the plant. The phloem translocate a variety of organic and inorganic solutes mainly from the leaves from from the leaves to other parts of the plant. 11.31 How do plants absorb water? We know that the root absorb most of the water that goes into plant. Obviously, that is why we apply water to the soil and not on the leaves. The responsibility of absorption of water and minerals is more specifically the function of the root hair that are present in millions at the tips of the root. Root hairs are thin-walled, slender extension of root epidermal cell that greatly increase the surface area for absorption. Water is absorbed along with mineral solutes by the root hairs purely by diffusion. Once water is absorbed by the root hair, it can move deeper into root layers by two distinct pathways. First, Apoplast pathway, second, Simplastic pathway. The apoplast is the system of adjacent cell wall that is continuous throughout the plant, except at the Casparian strips of the endodermis. In the roots, the apoplastic movement of water occurs exclusively through the intercellular space and the walls of the cell. Movement through apoplast does not involve crossing the cell membrane. This movement is dependent on the gradient. The apoplast does not provide any barrier to water movement, and water movement is through mass flow. As water evaporates into the intercellular space or the atmosphere, tension develops in the continuous streams of water in the apoplast. Hence, mass flow of water occurs due to the adhesive and cohesive property of water. The symplastic system is the system of interconnected protoplast. Neighboring cells are connected through cytoplasmic strands that extend through plasmodesmata. During symplastic movement, the water travels through the cells. Their cytoplasm intercellular movement is through the plasmodesmata. Water has to enter the cell through the cell membrane. Hence, the movement is relatively slower. Movement is again down a potential gradient. Symplastic movement may be aided by cytoplasmic streaming. You may have observed cytoplasmic streaming in cells of the hydrilla leaf. The movement of chloroplasts due to streaming is easily visible. Most of the water flow in the roots occur via apoplast since the cortical cells are loosely packed, hence offer no resistance to water movement. However, the inner boundary of the cortex, the endodermis, is impervious to water because of a band of suberized matrix called the Casparian strip. Water molecules are unable to penetrate the layer, so they are directed to volar regions that are not suberized into the cells proper through the membrane. The water then moves through the symplast and again crosses the membrane to reach the cells of the xylem. The movement of water through the root layers is ultimately symplastic in the endodermis. This is the only way water and other solutes can enter the vascular cylinder. Once inside the xylem, water is again free to move between cells as well as through them. In young roots, water enters directly into the xylem vessels and or tracheids. These are non-living conduct and so are parts of the apoplast. The path of water and mineral ions into the root vascular system is summarized in figure 11.7. Some plants have additional structure associated with them that help in and mineral absorption. A mycorrhiza is a symbiotic association of a fungus with a root system. The fungal filament form a network around the young root or they penetrate the root cells. The hyphae have a very large surface area that absorb mineral ions and water from the soil from a much larger volume of soil. that perhaps a root cannot do. The fungus provide minerals and water to the roots. In turn, the roots provide sugar and nitrogen containing compound to the mycorrhiza. Some plants have an obligate association with the mycorrhiza. For example, pinus seeds cannot germinate and establish without the presence of mycorrhiza. 11.32 Water movement of a plant. We looked at how plants absorb water from the soil and move it into the vascular tissue. We now have to try and understand how this water is transported to various parts of the plant. Is the water movement active or is it still passive? Since the water has to be moved up a stem against gravity, what provides the energy for this? 11.321 Root pressure. As various ions from the soil are actively transported into the vascular tissue of the roots, water follows its potential gradient and increases the pressure inside the xylem. This positive pressure is called root pressure and can be responsible for pushing up water to a small height in the stem. How can we see that root pressure exists? Choose a small soft stemmed plant and on a day when there is a plenty of atmospheric moisture, cut the stem horizontally near the base with sharp blade. Early in the morning you will soon see drops of solution ooze out of the cut stem. This comes out due to the positive root pressure. If you fix a rubber tube to the cut stem as a sleeve, you can actually collect and measure the rate of exudation and also determine the composition of the exudates. Effects of root pressure is also observable at night. Early morning when evaporation is low and excess water collect in the form of droplets around spatial opening of veins near the tip of grass blades and leaves of many herbaceous plants. Such water loss in its liquid phase is known as guttation. Root pressure can at best only provide a modest push in the overall process of water transport. They obviously do not play a major role in water movement up tall trees. The greatest contribution of root pressure may be to re-establish the continuous chains of water molecules in the xylem, which often break under the enormous tension created by the transpiration. Root pressure does not account for the majority of water transport. Most plants meet their need by transpiratory pool. 11.322 Transpiration pool. Despite the absence of a heart or a circulatory system in a plant, the upward flow of water through the xylem in plant can achieve fairly high rate up to 50 meter per hour. How is this movement accomplished? A long standing question is whether water is pushed or pulled through the plant. Most researchers agree that water is mainly pulled through the plant and that the driving force for this process is transpiration from the leaves. This is referred to as the cohesion tension transpiration pool model of water transport. But what generates this transpiration pool? Water is transient in plants. Less than 1% of the water reaching the leaves is used in photosynthesis and plant growth. Most of it is lost through stomata in the leaves. This water loss is known as transpiration. You have studied transpiration in an earlier class by enclosing a healthy plant in polythene bag and observing the droplets of water formed inside the bag. You could also study water loss from a leaf using cobalt chloride paper, which turned colors on absorbing water. 11.4 Transpiration is the evaporative loss of water by plants. It occurs mainly through stomata. Example stomata. Beside the loss of water vapor in transpiration, exchange of oxygen and carbon dioxide in the leaf also occur through these stomata. Normally, stomata are open in the daytime and closed during the night. The immediate cause of the opening or closing of stomata is a change in the turgidity of the guard cells. The inner walls of the each guard cells towards the pore or stomatal aperture is thick and elastic. When turgidity increases within the two guard cells flanking each stomatal aperture or pore, the thin outer walls bulge out and force the inner wall into a crescent shape. The opening of the stomata is also added due to the orientation of the microfibrils in the cell walls of the guard cells. Cellulose microfibrils are oriented radially rather than longitudinally, making it easier for the stoma to open. When the guard cells lose turgor due to water loss or water stress, the elastic inner walls regain their original shape. The guard cells become flaccid and the stroma closes. Usually, the lower surface of a dorsiventral, often dicotyledonous leaf has a greater number of stomata, while in an isobilateral, often monocotyledonous leaf, they are about equal on both surfaces. Transpiration is affected by several external factors: temperature, light, humidity, wind, speed. Plant factors that affect transpiration include number and distribution of stomata, percent of open stomata, water status of the plant, canopy structure, etc. The transpiration-driven ascent of xylem sap depend mainly on the following physical property of water: cohesion, mutual attraction between water molecule, adhesion, attraction of water molecule to polar surface such as the surface of tertiary elements, surface tension. Water molecules are attracted to each other in the liquid phase more than to water in the gas phase. These properties give water high tensile strength, that is, an ability to resist a pulling force, and high capillarity, that is, the ability to rise in thin tube. In plants, capillarity is added by the small diameter of the tertiary element, the tracheids and the vessel elements. The process of photosynthesis require water. The system of xylem vessels from the root to the leaf vein can supply the needed water. But what force does a plant use to move water molecule into the leaf parenchyma cell where they are needed? As water evaporates through the stomata, since thin film of water over the cells is continuous, it results in pooling of water molecule by molecule into the leaf from the xylem. Also, because of lower concentration of water vapor in the atmosphere as compared to the substomatal cavity and intercellular space, water diffuses into the surrounding air. This creates a pull that is given in figure 11.9. Measurements reveal that the forces are generated by transpiration can create pressure sufficient to lift a xylem sized column of water over a 130-meter high. 11.41 Transpiration and Photosynthesis, a compromise. Transpiration has more than one purpose. It first create transpiration pool for absorption and transport of plants. Second, supplies water for photosynthesis. Third, transport mineral from soil to all parts of the plant. Fourth, cools leaf surface sometimes 10 to 15 degree by evaporative cooling. Fifth, maintains the shape and structure of the plant by keeping cells turgid. An actively photosynthesizing plant has an insatiable need for water. Photosynthesis is limited by available water, which can be swiftly depleted by transpiration. The humidity of rainforest is largely due to this vast cycling of water from root to leaf to atmosphere and back to the soil. The evolution of the C4 photosynthetic system is probably one of the strategies of maximizing the availability of CO2 while minimizing water loss. C4 plants are twice as efficient as C3 plants in terms of fixing carbon dioxide, making sugar. However, a C4 plant loses only half as much water as a C3 plant for the same amount of CO2 fixed. 11.5 Uptake and transport of mineral nutrients. Plants obtain their carbon and most of their oxygen from CO2 in the form of in the atmosphere. However, their remaining nutritional requirement are obtained from water and minerals in the soil. 11.51 Uptake of mineral ions. Unlike water, all minerals cannot be passively absorbed by the roots. Two factors account for this. First, minerals are present in the soil as charged particle which cannot move across cell membrane. Second, the concentration of mineral in the soil is usually lower than the concentration of mineral in the root. Therefore, most minerals must enter the root by absorption into the cytoplasm of epidermal cell. These need energy in the form of ATP. The active uptake of ions is partially responsible for the water potential gradient in roots and therefore for the uptake of water by osmosis. Some ions also move into the epidermal cells passively. Ions are absorbed from the soil by both passive and active transport. Specific protein in the membranes of root hair cells actively pump iron from the soil into the cytoplasm of the epidermal cell. Like all cells, the endodermal cells have many transport protein embedded in their plasma membrane. They let some solutes cross the membrane, but not other. Transport protein of endodermal cells are control points where a plant adjusts the quantity and types of solutes that reach the xylem. Note that the root endodermis, because of the layer of suberin, has the ability to actively transport ions in one direction only. 11.52 Translocation of minerals. After the ions have reached xylem through active or passive uptake or a combination of the two, their further transport up the stem to all parts of the plant is through the transpiration stream. The chief sinks for the mineral elements are the growing region of the plant, such as the apical and lateral meristems, young leaves developing flower, fruits and seeds, and the storage organ. Unloading of mineral ions occur at the fine vein ending through the diffusion and active uptake by these cells. Mineral ions are frequently remobilized, particularly from older sensing parts. Older dying leaves export much of their mineral content to younger leaves. Similarly, before leaf fall in deciduous plants, minerals are removed to the other parts. Element most readily mobilized are phosphorus, sulfur, nitrogen, and potassium. Some elements that are structural component like calcium are not remobilized. An analysis of the xylem exudate shows that though some of the nitrogen travels as inorganic ions, much of it is carried in the organic form as amino acids and related compounds. Similarly, a small amount of P and S are carried out as organic compound. In addition, a small amount of exchange of material does takes place between xylem and phloem. Hence, it is not that we can clearly make a distinction and say categorically that xylem transport only inorganic nutrient while phloem transport only organic material, as was traditionally believed. 11.6 Phloem transport flow from source to sink. Food, primarily sucrose, is transported by the vascular tissue phloem from a source to a sink. Usually the source is understood to be that parts of the plant which synthesize the food, that is the leaf, and the sink the part that need or store the food. But the source and the sink may be reversed depending on the season or the plant's need. Sugar stored in roots may be mobilized to become a source of food in the early spring. When the buds of trees act as sinks, they need energy for growth and development of the photosynthetic apparatus. Since the source sinks relationship is variable, the direction of movement in the phloem can be upward or downward, that is bidirectional. This contrasts with that of the xylem, where the movement is also unidirectional, that is upward. Hence, unlike one way flow of water is transpiration, food in phloem sap can be transported in any required direction so long as there is a source of sugar and a sink able to use, store or remove the sugar. Phloem sap is mainly water and sucrose, but other sugars, hormones, and amino acids are also transported or translocated through phloem. 11.61 The pressure flow or mass flow hypothesis. The expected mechanism used for the translocation of sugar from source to sink is called the pressure flow hypothesis. As glucose is prepared at the source, it is converted to sucrose. The sugar is then moved in the form of sucrose into the companion cell and then into the living phloem sieve to cells. By active transport, this process of loading at the source procedure by hypotonic condition in the phloem, water in the adjacent xylem move into the phloem by osmosis, and osmotic pressure builds up the phloem sap will move to areas of lower pressure. At the sink, osmotic pressure must be reduced. Again, active transport is necessary to move the sucrose out of the phloem sap and into the cell, which will use the sugar, converting it into energy, starch or cellulose. As sugars are removed, the osmotic pressure decreases and water moves out of the phloem. To summarize, the movement of sugars in the phloem begins at the source where sugars are loaded into a sieve tube. Loading of the phloem sets up a water potential gradient that facilitates the mass movement in the phloem. Phloem tissue is composed of sieve tube cells, which form long columns with holes in their end walls called sieve plates. Cytoplasmic strand pass through the holes in the sieve plates, so forming continuous filaments. As hydrostatic pressure in the sieve tube of phloem increases, pressure flow begins and the sap moves through the phloem. Meanwhile, at the sink, incoming sugars are actively transported out of the phloem and removed as complex carbohydrate. The loss of solute produces a high water potential in the phloem, and water passes out, returning eventually to xylem. A simple experiment called girdling was used to identify the tissue through which food is transported. On the trunk of a tree, a ring of bark up to depth of the phloem layer can be carefully removed. In the absence of downward movement of food, the portion of the bark above the ring on the stem becomes swollen after few weeks. This simple experiment shows that phloem is the tissue responsible for translocation of food, and the transport takes place in one direction, that is towards the root. This experiment can be performed by you easily. Summary: plants obtain a variety of inorganic elements and salts from their surrounding, especially from water and soil. The movement of these nutrients from environment into the plant as well as from one cell to another plant cell, essentially involving movement across a cell membrane, transport across Transport across cell membrane can be through diffusion, facilitated transport, or active transport. Water and minerals absorbed by roots are transported by xylem, and the organic material synthesized in the leaves is transported to the other parts of the plant through phloem. Passive transport, diffusion, osmosis, and active transport are the two modes of nutrient transport across cell membrane. In living organism, in passive transport, nutrients move across the membrane by diffusion without any use of energy, as it is always down the concentration gradient and hence entropy driven. This diffusion of substance depending on their size, solubility in water or organic solvent. Osmosis is the special type of diffusion of water across a selectively permeable membrane, which depends on pressure gradient and concentration gradient. In active transport, energy in the form of ATP is utilized to pump molecule against a concentration gradient across membrane. Water potential is the potential energy of water molecules which help in the movement of water. It is determined by solute potential and pressure potential. The osmotic behavior of the cell depending on the surrounding solution. If the surrounding solution of the cell is hypertonic, it gets plasmolyzed. The absorption of water by seeds and the dry wood takes place by a special type of diffusion called imbibition. In higher plant, there is a vascular system comprising of xylem and phloem responsible for translocation. Water, minerals, and food cannot be moved within the body of a plant by diffusion alone. They are therefore transported by a mass flow system, movement of substance in bulk from one point to another as a result of pressure differences between the 2 points. Water absorbed by root hairs move into the root tissue by two distinct pathways, that is apoplast and symplast. Various ions and water from soil can be transported up to a small height in stem by root pressure. Transpiration pool model is the most acceptable to explain the transport of water. Transpiration is the loss of water in the form of vapor from the plant parts through stomata. Temperature, light, humidity, wind speed, and number of stomata affect the rate of transpiration. Excess water is also removed through strips of leaves of plants by guttation. Phloem is responsible for transport of food, primarily sucrose from the source to the sink. The translocation in phloem is bidirectional. The source-sink's relationship is variable. The translocation in phloem is explained by the pressure flow hypothesis. Thank you friends for listening this audio, and please post your comment if you want any kind of improvement, and just stay tuned with Biology Critic and enjoy the knowledge. Okay, bye.